And then I'll do a, thank you. So I'll just do a brief introduction. And so the way we're going to do the questions, if anyone has them during the talk, and welcomes them to be asked in the chat as the slide is up, and I'll just interrupt and ask the question right then and there. So, all right. So today's seminar speaker is Professor Anne Draelos, and she's an assistant professor in biomedical engineering and computational medicine and bioinformatics at the University of Michigan. And her lab is focused on machine learning and statistical techniques to facilitate real-time analysis of high-dimensional neural and behavioral data. And Anne got her PhD in physics and did postdoctoral research in neuroscience at Duke University. So thanks for being here today. And when you're ready, take it away.
All right. Yeah. Thank you so much for the invitation. I think the A3D3 is a pretty unique and relevant, obviously. I've also enjoyed looking at some of the previously recorded seminars. So like Brian mentioned, I work on sort of real-time modeling with adaptive interventions geared towards neuroscience experiments. So in general, my work is motivated by the question of how to construct and test new hypotheses, given the increasingly high dimensional spaces that our data live in. So for those of us who are in neuroscience, we're all really aware of this recent, really rich data explosion, as we can increasingly record from more and more neurons in the brains of live animals. So for instance, with using optical imaging techniques, we can record up to entire brains in the zebrafish larva, about 100,000 neurons. And there's also been some advances recording in the mouse brain up to about 10% of it. But it's not just the increasing neuron dimension that presents us with high dimensional data challenges. The dimensionality of our behavior spaces, so the sort of external metrics that we observe, the time scale or resolution at which we can measure various behaviors like the kinematics or vocalizations, those are all increasing, as well as the external stimuli. So the environment in which our model organisms are living, the kinds of, for instance, here in these images, visual stimuli that we present, moving towards naturalistic or even hypo-realistic stimuli that evoke strong neural responses. These are all high dimensional. And what we'd like to do is be able to make provable claims about, for instance, how many thousands or hundreds of neurons are working together in concert in response to some sort of complex external stimuli that are evoking fine-grained behavior. The time that we have to conduct experiments that are directly and ideally causally testing our hypotheses about how these complex things interact, that time has remained the same. There are only 24 hours in a day. So we can't measure neural or behavioral responses to, all possible visual inputs. We can't test all possible ways to intervene in the neural system or during behavior. So how do we choose? And so the ideas that we have for tackling that problem of scaling experiments into these higher dimensional spaces is to use adaptive experimental designs. So you have all probably used an adaptive design to play one or more of these games. In each case, we conduct some sort of guess or trial or test, and we receive an answer about how well we did. So in Wordle, we receive black, yellow, or green squares. And then in Battleship in 20 questions, it's much more of a yes, no, hit, miss kind of. But the key thing is that each time we receive feedback, we're able to better design the next question that we ask. So for instance, if we look at the game of 20 questions, it's my job to think of an object, and it's your job to ask me yes or no questions to figure out what I'm thinking of. And given the space of semi-infinite possibilities of what I might be thinking of, this is still a winnable game for you because you hear the answer to your question each time you ask, and you can therefore modify which question to ask next. So if, for instance, I were thinking of a duck, you could solve this by asking a series of questions like, is it a mammal? Does it have wings? Does it have a beak? And so on. But what if instead I forced you to ask all of your questions at once without hearing any of the answers to them? You could write down a list of questions to try to figure out a duck, but only if you sort of had that prior in your head. You sort of knew what I was thinking of in advance. You would need potentially thousands of questions to cover every possible object I might be thinking of, assuming I wasn't thinking about said duck. Right, so the key thing is that each time you ask a question, you immediately receive an answer, which allows you to intelligently pick the next question based on that answer. So this notion of an adaptive experimental design allows us to take advantage of data as we collect it. Furthermore, rather than predetermining all experimental parameters of interest ahead of time, we can instead leverage adaptive designs to update our model, change where we look, change other stimulation parameters, and so on and so forth, while the experiment is ongoing. So in the talk today, I'll focus on two topics. The first tackles the problem of learning and predicting neural data with real-time models. And the second deals with a couple of ways in which choosing optimal interventions allows us to efficiently discover something about neural circuits that would otherwise be too costly time-wise. And you can think of these two things by analogy as understanding the answers the brain is giving us and designing the next best question to ask based on those answers. All right, so first we're going to think about how to design models that learn in real time using various neural or behavioral data streams and how to do this experimentally. So experimentally in the lab, we are relying on a variety of neurotechnology tools to both observe and perturb behavioral or neural activity. Sort of think of any complex combination of these things if you want, people are studying it. And so what we want is to incorporate our models into these kinds of experimental pipelines to provide real-time quantitative descriptions or inferences for the system under study, and to help generate new testable hypotheses on the fly, right? So plug it in order to make it this closed loop kind of feedback system. But actually constructing experimental designs that draw from most, if not all of these elements is really difficult. And frequently implementations of these experiments, especially those with real-time constraints, are very lab specific and difficult to generalize, in part because of the tight integration required between all of these components. So what I did during my postdoc, and I'm sort of continuing on now as a professor, is working on a very flexible software platform written in Python, which is the language of choice for many, if not most, neuroscientists, that we called Improv, which allow us to engineer real-time experiments. So Improv is going to handle all of the complex communication between the various experimental components, as well as provide the interface to the model, which is my personal favorite aspect of this, so that we can, use this model to help us understand the data streams that we have, so capture some sort of information about it, and then suggest how we're going to change the experiment next. So let me give you a concrete example of integrating our real-time models into an experimental setup. This is work done in the lab of a collaborator, Eva Naumann at Duke University. Her lab focuses on the optomotor response in larval zebrafish. So this is just an example of the kind of neural activity, so the neural data that we're going to be collecting using 2-photon calcium fluorescence imaging. This is a single plane through the brain focused on the optic pretectum. And one of the reasons to study this region is its involvement in this highly reproducible behavior. So if you show the fish a series or a set of moving gratings underneath the fish will swim to align themselves to the visual motion. So if you showed it a leftward moving grating, the fish would want to turn and swim to the left, rightward moving grating and swim to the right, and so on and so forth. So this is a very robust thing to study. So if you want to do this in reality, it sort of involves a lot of steps if you care about trying to provide quantitative descriptors for this analysis. So we saw we can get brain images, we can get those in real time fairly easily. But we also need to do things like motion correction. We need to figure out, okay, here's a neuron. We want to extract a time trace. That's this sort of orange line here. And then potentially we may also want to deconvolve to get neural spikes, which are more of the sort of thought to be the unit of computation. So you can do these kinds of things, especially with the help of an algorithm called Cayman Online. But really integrating each of these parts and getting things to run really quickly is a little bit of a challenge. And this is what we used Improv for. So each new frame of data, you have to analyze it by itself. And this is a streaming setting. And you can't leverage future information because you don't have it. So using Improv, each sort of step that you need to process this data is considered a single actor in this setup. So we're running each of these steps in parallel or concurrently rather than sequentially, which allows us to stay at real-time speeds. So we're going to process the image using this Cayman online algorithm. We're going to take information about what sort of visual stimuli we've been showing the fish. We're going to correlate those two timing, and we're going to feed it into, in this case, for example, a sort of simple model that's commonly used in systems neuroscience. So it's called the linear nonlinear Poisson model. Effectively, what it's looking at is trying to understand the influence of some sort of baseline firing rate for each neuron, some sort of information about how the neuron is going to respond to the visual stimulus, information about how recently it fired, as well as information about connections from other neurons in this highly connected system. One of the nice things about this particular model that we've modified to work in the streaming setting is that it is convex. And so for convex problems, we're able to use things like stochastic gradient descent. And the convergence to a global optimum of this model is guaranteed with sufficient data, even if you fit it one piece of data at a time. So we're effectively guaranteed to achieve the same result when we fit the model in real time as when we fit the model using offline data all at once. So just to give you a little example, this is how we've set it up in the lab. We have raw images collected here. And then in less time than it takes to acquire an image, we're presenting this process frame here. So we've done all of the analysis in less time than it takes to acquire a single image. So here, what we're showing is a backward moving grading. So that's the purple downward arrow. And you can immediately see which neurons are responding to backward motion. We might show a forward right stimulus here in yellow, and we'll see different neurons. And as we show more and more stimuli, we can begin to build up a picture of up to the moment information about how each neuron across this field of view is responding. And here we've color coded each neuron based on how it responds most strongly to different visual stimuli. Now, again, we're running a lot of this stuff concurrently. So there's nothing to stop us from doing other model fits. And here, like I mentioned, we're going to use things like stochastic gradient descent to fit a particular model to this data to help us also estimate functional connections between the neurons, visualized here by these green lines. So with this platform, we can analyze in real time and obtain functional types of each of these neurons across our field of view, which is something that you cannot know a priori. So in order to get this information, you must measure it. And so here we're able to provide the analysis effectively simultaneously with the measurement. And as I mentioned, we're fitting this model. And what we find is that the model fit, the log likelihood here, that of the model that we fit online converges to that of the model fit that we obtain using all of the data. You can see that it converges here within maybe 3.5 or 4 minutes worth of data. So what's relevant about this is that this is after only showing one set of visual stimuli, so one set of experimental parameters. And it's much more typical to, of course, go through this 5 times, 10 times, right? You're averaging the responses that you get to have a more robust picture. But if you have good estimators of uncertainty in your model parameters, you can first fit online and choose to stop early, right? So here's a point where we could have chosen to stop. We have sufficient certainty about what we see, and we can then immediately move on to the next phase of the experiment. All right, so what I just told you about uses a sort of static model where you just want to find good parameters, but you already know the structure of the model right off the bat, right? Those are those 4 parameters sets that I listed off. So next I want to look at a slightly harder case where we might want to intervene or perturb neural activity based on neural dynamics, not just the semi-static functional properties of individual neurons. And so one way that people think about getting a handle on high dimensional neural data is to instead look at the neural data using a low dimensional representation. So there's been a lot of work on these so-called population level models. They've been very successful in describing the coordinated activity of larger populations of neurons. So here, for instance, we have 3 neurons, and we're going to get their spike trains and plot them in this 3-dimensional space. And one of the goals of the low dimensional representation is to find a lower dimension, in this case 2, that can still well describe the the neural activity across time. And so we call this sort of a neural trajectory. It's been a really powerful technique, very successful, particularly in motor cortex. For instance, plotting the first 2 dimensions of the neural subspace, we see these nice rotational dynamics that well align with the hand kinematics recorded simultaneously during a reaching task. So these models are very exciting, very interesting, but they're learned offline. And they are typically imposing some sort of assumption about the underlying dynamical system that is generating these trajectories. So for real-time and adaptive experiments, we might instead want to learn what these trajectories are as we go, so that we can then perturb them at specific points along the trajectory within the identified neural manifold. So to start with, we needed a method of dimension reduction that works on real-time data streams. So there are versions of streaming or incremental SVD, but the vectors that they return are quite variable throughout the time series of data and can abruptly flip or change in a way that makes it difficult to do anything with, to build another model on top of. So instead, we started from a streaming version of SVD by Baker et al. And rather than choosing the top K singular vectors during the SVD, which is quite traditional, we instead chose to minimize the change in the singular subspace bases between data points, so between QT and QT minus 1. This is an orthogonal Procrustes problem with a known solution and can be computed really efficiently. And what we find is that if you look here at this plot, the derivative of the identified basis vectors drops over time. And after maybe 8 to 10 seconds, we come to a time-stable, low-dimensional representation of the data. So this gives us a really stable foundation, right? That low-dimensional subspace, and it works really well on many kinds of high-dimensional data, time series data. All right, so suppose now that we have that data in the low-dimensional space, for instance, data trajectories traced out in gray here. Our next goal is to figure out how can we predict what happens. Having seen this data, we'd like to know what happens in the next second. So the way that we did this is we're using a mixture of Gaussians to identify the locations of the data, where here the black dots are the mean of the bubble, and this orange is the variance plotted out to 2 sigma. And then once we have these tiles sort of wrapping, or bubbles wrapping the data, we can also learn a transition matrix between the tiles using a hidden Markov model. So for instance, for this kind of trajectory, starting here and going over to here, we might learn this series of jumps. Notice that we're not focused on learning an exact dynamical model of the data, just an approximate one. So we're learning a sort of coarse tiling within the space, along with a set of predictions for transitioning between those tiles in space. So no sort of parametric assumption has to be made about the neural dynamics themselves. And this allows us to stay flexible to trajectories that change over time. Because as pretty as some of this data is, in reality, if you're doing it in real time, it changes a lot. So we need to be able to remain flexible due to evolving trajectories, even due to noise, and allow us to capture multimodal probability flows, like when trajectories are splitting. So here's a quick movie showing this method work in simulated data, where the gray is the dimension reduced noisy data, and the oranges are the algorithm figuring out where are the data, and then building a transition matrix that sort of shows this evolution of the trajectory in a circle. So we're going to evaluate this method by looking at 2 quantities. The first is the log predicted probability, which is the probability of predicting data some number of time points into the future based on all the data that we've seen so far. And second, we looked at the entropy of the predictive transitions. So this shows us how much predictive information that we've managed to capture about the data. The dashed line up here is just the max entropy, which is log 2 of the number of tiles used. Each of these quantities can also be computed very efficiently, which lends itself to real-time work. All right, so if we go back and look at some of those rotational dynamics that I showed you earlier, here each blue is tracing out a trial. And you can see as we observe more and more trials, we learn a better covering of the space and better prediction of what's going to happen next based on where we are. And you can see there, let me just go back really fast. up in the top right corner, we're going to see something surprising. So a trial that doesn't match, yep, there it is, any other trial that it's seen so far. But we're able to quickly adjust to this new information and return to our ability to predict. So if we look at the log predictive probability, you can see this going up quite nicely and the entropy going down, along with a few surprising events in the data. And the data itself are showing these same kind of organized rotations, which the model learns really effectively. So I think I told you at the beginning of this that this works in general on higher dimensional data across time. So there's no reason we have to use it on neural data. So what we did is we actually looked at some behavioral data. So this is just a video of a mouse's face, which I'll actually show you a little bit later. And without knowing anything about what was in the video, we can just take the pixel dimension, which is high dimensional, and find a lower dimensional representation of it. So here, you can see the data have a much more complicated, somewhat less structured form, which is harder to model. But we're still able to get a handle on these coarse sort of transitions up and down in this lower dimensional space as the mouse is in fact doing a decision-making task. So like I said, it's a little bit more noisy, but we're still able to capture some coarse structure and extract predictive information as shown by this clear drop in entropy. So we apply this to a variety of data sets. The full details are in the Neuros paper that we published on this. But basically, you can see that the low dimensional representation of the data can take on different shapes. This is a URAP representation. But in each case, we're able to learn something about the neural data independent of the way that it was acquired. Okay, now returning to experimental integration, our model species of choice here is this larval zebrafish. Like I showed you earlier, we're able to get some functional characterizations of the neurons. Now let's look at all of these neurons at the same time and find that low dimensional representation while it's doing this task of looking at moving gratings. Yep. So as we see different gratings, and this movie is slightly sped up, we can see that we're able to start to capture some sort of information about these loops that we're beginning to see, as well as occasionally having surprising ones. And if you take the entire data set and you color it at the end, we see this nice correspondence with what stimuli the FIS was being shown at the time while we were acquiring the data. So for instance, here it was seeing rightward motion, and here it was seeing mostly backward motion, and you can see some sort of nice splitting there. That is not built into the model, it just came out of it. And one of the things that I think would be interesting to work at is because we have these data trajectories and the prediction ahead in time, we can think about, okay, if I know I'm about to come to some sort of inflection point, let's go ahead and design a perturbation. Let's stimulate the neurons and start to test our understanding of how these trajectories are evolving. Yeah, so we benchmarked a lot of this stuff to get it to run on, ordinary workstations that a lot of neuroscience labs could have. The actual time to predict some steps ahead into the future, rather than the time to really construct the model, is running at faster than 10 kilohertz, which makes real-time closed-loop experimental designs very feasible. And just to sort of give you one quick example, if you plot out the sort of predictions ahead in time, looking at this graph over here, the log predicted probability, of course, we're going to do slightly worse as we predict further and further out in time, especially given the noise level of the data. But this is actually quite good and very sufficient for designing perturbations at projected trajectories ahead in time. Yeah. Okay, so one other immediate way that we can use this identified stable subspace. So previously I showed you that we can use current neural data to predict future neural data, and then think about ways to design perturbations, which I'll get to in a minute. We can do the same thing with behavioral data. Use current behavioral information, latent behavioral information, to predict future latent behavioral information. And there's no reason we can't do current neural data to predict future behavior and vice versa. So what we did is we looked at an analysis from this paper, Musal et al., where they have simultaneously recorded behavior video alongside 2-photon neural data. That's these traces here. And the original paper looked at this question of how to predict neural activity from behavior features. So we're trying to identify beta here. And so what we did is we looked at the low dimensional representation of this behavior video and tried to predict the simultaneously occurring neural activity. And what we find is that, so this is just the same video I showed you a little bit earlier, the behavioral trajectories within the latent space. We're able to quickly identify regressors. And then if we project them back up onto that higher dimensional space that is the original image, coloring them by the strength of the regressor, we can basically highlight regions that are highly predictive of the neural activity. It's basically saying, these are the features of behavior that you should be measuring if you're looking for neural correlates. And so here, for instance, we might say that we're going to image where the nose is, but we may also want to pay attention to these more fine cheek movements, which is in fact a result that recapitulates the finding from the original paper. And we identify this within only a few minutes worth of data. And since we can do this in real time, we could think next about trying to causally link neural manipulations to behavioral outcomes or vice versa. So let's do that. Now that we have these kinds of real-time neural models, complete with predictive information, we can consider closing the loop and designing new ways to perturb neural circuits. So one of the ways that we looked at doing this is to use perturbations or direct neuronal stimulations to learn something about the functional connections among the neurons. So learn something about the network structure of the neurons. One of the reasons we first got interested in this, especially in Eben Amman's lab, is because there are techniques that exist, like three-dimensional holographic photostimulation, that have made it possible to stimulate individual neurons as well as groups of individual neurons anywhere in a three-dimensional volume. So this is basically unprecedented access to the brain. And we want to think about how we would design optimally some sort of stimulation pattern that lets us rapidly learn something about the neural circuit. But the zebrafish has, tens of thousands up to 100,000 neurons that we can access. So how do we even go about designing experiments to probe the connectivity of that very large network? A sort of naive approach would be to stimulate every neuron that we can access and observe its neighbors' responses. So for a network of 100 neurons, this could take you about an hour, which is totally doable. For 1,000 neurons in your neural circuit, this could take you maybe a whole workday. But if you tried to map the possible 100 million connections in a network of even 10,000 neurons, it's very infeasible, requiring 3 days of constant photostimulation, at which point you have fried your fish brain anyway. Right, so that's not going to work. So instead, we turn to the statistical framework of group testing, which probably more of you have heard of now, given the COVID pandemic. So you take samples from individuals and you pool them into a group to test a single time, rather than having to test each person individually. You could see how this is much more statistically efficient than trying to look at everyone individually. So if it's negative, you've cleared a lot of people, and if it's positive, then you just repool with different groups. And based on subsequent test results, you can identify who's infected. So leveraging this, we're going to apply it to this problem of trying to figure out who's connected to this neuron. So if I'm looking at this particular neuron and I'm stimulating some subset of its potential connections, based on if we see a response or not from this neuron, we're able to say, okay, then at least one of its neighbors got stimulated. And note that here we're just addressing the sort of existence of connections, or the binary network problem. We're not yet interested in the strength of these connections. And what's particularly helpful about this kind of method is that we can parallelize our observations across all neurons. So during the things like 2-photon calcium fluorescence imaging, you're imaging all the neurons at once. So the math scales really efficiently from looking at one neuron to looking at all possible neurons, maybe not neurons that you stimulated. So if we look at the math for just a single neuron at a time, like I said, if the neuron is stimulated, X, and if it is connected to the observation neuron, W, then you get a 1. So this is basically an OR operation across all its connections and stimulated neurons. Like most things in biology, our measurements are noisy. So we're going to include false positives and false negatives in the model for what we observe. And if you work through the math here, looking at the log likelihood and trying to maximize this, you find that there are 2 values that your response could take. Yes, the neuron responded. No, the neuron didn't. And this turns into basically a bit matching problem. So was my connection stimulated? Did I respond? And you want those things to be the same, well aligned. The bit matching problem in general is NP hard. So instead we created a new relaxation of the problem. And rather than considering just binary results, yes, it responded, no, it didn't, we're going to consider a slightly more continuous representation on the response we saw, as well as on the connections that we find, and just relate them to each other via constraints. But it's still just the basic binary network problem. The nice thing about structuring it this way is that it turns into a bipartite graph structure, which allows you to decompose this problem. So A is, what was the response, and W is there a connection? And if we use dual variables to account for the original constraints, we have this basically perfect dual decomposition, which basically just means for really, really efficient optimization that scales really well, even with increasing number of neurons in the network. This is just looking at a quick non-random network where here we're going to be stimulating the network using the single neuron at a time method, and here we're going to be using our new group testing method. Black means there's no connection, white means we think there is a connection, and gray means we're sort of uncertain, and we have uncertainty built into this model. So you can see the kinds of speedups that we get with group testing for this particular simulated network of 150 neurons. You can see that the group testing approach is faster, requires a fewer number of tests, and is ultimately less noisy than that naive method. To evaluate this quantitatively, we looked at specificity and sensitivity. The blue line here is our group testing method, whereas the green line is naive, and we want both of these values to be close to 1. And so you can see that group testing does this within a few hundred tests for a network of 1,000 neurons, whereas the naive method is going to take many more tests. The dotted lines here just show different test error rates. What we're really interested in is how we scale this. And so you can see here, as we grow the network from 100 neurons up to 10,000 neurons, it scales really well, requiring just about a quarter of the number of neurons for tests. So the number of tests you have scales with the size of your network in a really consistent fashion. And I should note here that what we're doing is not using random groups, which is the sort of typical group testing approach. We're actually being a little bit more clever. So we're using the results of each stimulation, updating our model in real time, and then saying, based on current uncertainties in the network that our model is seeing, let's stimulate the most uncertain connections. Basically, this allows us to target our uncertainty and rapidly determine the entire network. This is also this kind of problem, determining the structure of a 10,000 neuron network isn't necessarily possible to even compute offline without a large cluster to hold everything in memory. But it's not necessary. You can see here that we're able to do this, again, in real time because we're using this iterative approach. And here our method can update after the results of a stimulation in less than 2 seconds, really pushing that real-time inference on large networks. Okay, and then lastly, I'm going to talk about how we can incorporate external stimuli into the loop. So I think in the beginning, I was telling you about some neural data, some behavioral data, and some external stimuli. So here, rather than looking at direct neuronal perturbations, let's look at how we can drive neural activity indirectly, still in a closed loop format. So going back to the optomotor response in our larval zebrafish, the typical kind of characterization of this neural circuit involves showing the fish 8 different directions of moving gratings underneath it. So this is effectively like a one-dimensional kind of problem. And it's very possible to basically show all 8 gradings, show it 3 or 4 times in a single experiment, and have really robust results. But it's kind of a coarse measurement, and we're really interested in looking at much finer detail in the neural response properties. So what we did is we wanted to look at, okay, let's make this a 2-dimensional problem. It's not really high dimensional quite yet, but even just going up to 2 dimensions, I'll show you, can be somewhat difficult. So if we vary the direction of the moving grading that we show to each eye, and we look at slightly finer angular resolution, so rather than 45-degree intervals, we're looking at 15-degree intervals, which the neurons are sensitive to. This presents us with a matrix of possibilities of close to 600 distinct stimuli. So now we're getting to the point where 600 stimuli, while possibly doable, is not really doable. Many, many hours. The fish doesn't necessarily sit under the microscope well that long. So we need something other than a brute force approach. And in our case, we turn to online Bayesian optimization. So the way this works, okay. So the way this works, we can look at one neuron at a time, but like many things of what I've just shown you, in fact, we can do this in parallel, but we'll focus on one neuron at a time for now. We can show the fish some stimulus, so 210 degrees in the right eye, 345 in the left eye, and we see the fluorescence response, so basically the neural activity, as a response to this stimulation. Using a small subset of these initial activity curves, we're able to, with a Gaussian process, fit an estimate of the neural tuning curve. So in 2 dimensions, the color here is the magnitude of the response that we expect. And you can see it looks like a 2-dimensional Gaussian bump, which is what we would expect. So that's sort of our prior. We also get uncertainty in our estimation of the tuning curve. Now, using a combination of the tuning estimate itself and our measure of uncertainty, so we're going to trade off between explore and exploit to pick the next best stimulus to display based on current information. So you can see here as we, let's see, we may have sampled here. Let's go ahead and sample over here now. And you can see our neural response has increased, right? And so you just do this again and again. You can iterate until you've reached some chosen confidence level in your tuning curve estimate. And then, for instance, look at the next neuron. So here's an example of how this works. So we see each of these fluorescence traces, which again, we can get in real time, and use this to update our model. And then using the tuning curve estimate and the current uncertainty, basically say, okay, what's the next best stimulus we should be presenting to this fish? We did this sequentially. So we took the first neuron, optimized it, and then the second neuron, optimized it, and the third, and so forth. And in practice, since again, we can record from all neurons at the same time, each new neuron that we optimize comes with free information from the previous trials. So it takes very few stimuli to progressively optimize larger populations. Yeah, so in order to validate this kind of method, we did the somewhat more brute force approach, which required about 144 stimuli. Still not anywhere near 600, but 144 was doable. You can see here that the GP fit, the Gaussian process fit here to our data agrees very well with the kind of real-time GP fit, which only used 8 stimuli versus 144 stimuli. Right, so if our goal is really to learn where the peak value of our responses are, I would say that this does really well. And like I mentioned, we can optimize a large number of neurons with a few number of stimuli. So we found that only each of these lines is a different fish or trial, imaging session. And on average, you can optimize 300 neurons or more with only 15 stimuli. So that's about 7 minutes worth of data collection using real-time adaptive methods versus many, many hours using non-adaptive methods. And because we're modifying which stimulus we pick based on those real-time responses, the algorithm adapts itself to different neural population structures. So if we look, for instance, here, this data was recorded from the optic pretectum. And what we find is, looking at this heat map, that most of the neurons prefer whole field motion. So the same direction of moving grading in each eye. That's why it's on the diagonal here. Whereas looking at the optic tectum, we found some regions where, yes, we still care about whole field motion, but there's also two other groups within the population that prefer converging or diverging sets of motion per eye. And what's great is that these white X's show us where the algorithm had chosen to sample. So based on where you have responses in your neural population, this is where you should choose to sample. You should obviously have a few samples outside of that, but you can see that the algorithm is adapting itself to different neural populations. So you're not going to waste time, having to do this entire grid search, having to cover all of your bases. Instead, you can stay adaptive and pick up the structure that's actually in your neural population. All right, so I hope I've shown you that one particularly powerful way to get at these more causal interventional experiments that can efficiently probe up to even entire brains in the future is to use adaptive experimental designs. In particular, I've talked about 3 key components to these designs: methods and a platform to enable real-time data analysis that give us those current estimates of brain state, flexible neural models that we can build in real time while the experiment is running and still be adaptable to the changing dynamics across time. And lastly, algorithms that also run in real time to help guide our choice of next intervention. So some sort of online optimization routine, so that we can really perturb systems in more complex ways and really explore these higher dimensional stimulus or parameter spaces. All right, so this work was, of course, done in collaboration with a number of excellent people in the labs of my postdoc mentors, John Pearson, as well as Eva Naumann, and some external collaborators. Thanks also to our funding sources. And if you want to check out the kind of code that we use, it's all here, Project Improv in particular. We've been able to, beyond what I've talked about today, integrate with other code bases and other experimental setups, and are interested in making adaptive designs accessible to everyone in neuroscience. So if you're interested, please reach out. And thanks, I'll be happy to take any questions.
Well, thanks, Anne. So if there are any questions, feel free to raise your hand and I'll call on you and you can unmute and ask. And if there aren't any right away, then I had one I was wondering about. So I'm an astronomer, so I definitely don't know all the neuroscience details, but I was wondering, you mentioned, so this is for the binary problem of just kind of identifying the connections. And I was curious about what kind of changes, what kind of added complexity is involved in figuring out like the strength of the connections, like you said, and how would that change things?
Yeah, good question. So we're showing actually that if you take this binary problem and extend it into the space where you're looking for the strength of connections, you're really hitting more of a compressed sensing kind of approach that people have previously studied. I don't know if you're familiar with it. And so there is a way to basically relate those two things. But I'd say just practically, one of the issues with determining strengths of connections is that in a lot of neural populations, the majority of the connections that exist are very small synaptic weights. And so designing ways to tease out more of the signal from the noise is something that's quite interesting. Our approach, we did account for some test error rates on the outcome of the stimulation. And I'm working now on a slightly more biologically plausible model looking at the input to the system. So if you're unsure how you've stimulated, how this would affect things. But I think the binary method approach would be great for establishing, basically narrowing down your search space initially to the existence of connections, and then using repeated stimulation to get at how strong are these connections.
Nice, thanks. I see we have a question from Eli.
I was muted. Yeah, thanks. Thanks, Anne, for joining us and for a great presentation. Really enjoyed it. I wanted to ask a general question in terms of applying these methods of dimension reduction or building a neural representation from data and then active based on that in online fashion, adaptive fashion, requires some filling some constraints in terms of the response time, right, and the compute time that this method will have to adhere. So I wanted to ask about more perspective on this, whether the particular method would be chosen kind of like as a balance between the accuracy, right, and the time that it can be useful in terms of eventually being more optimal at achieving the adaptive task.
Yeah, no, it's a great question. So we didn't explicitly look at the trade-off between time and accuracy, but I'll tell you a couple things. We did look at sort of extensive timing, and one of the things that we really wanted is to have this work on a fairly standard workstation for other people to use. We are using a single GPU to help boost some of these calculations in real time because you can do them in a parallel fashion. In terms of accuracy, so one of the things you can change in the model is the number of nodes that you're using, so the number of tiles that you're using to look at your space. So you would assume that if you wanted to be more accurate, you would have more tiles on your space so that you could have finer grained transitions. And what we found is that, yes, you can give it more nodes, for instance, 20,000 nodes versus 1,000 nodes. And obviously you are slower if you have more nodes. It's scaled n squared. But what we found is that even if you gave it lots of nodes and you included them in the computation, you're really not using most of those for prediction. So if you look at the tiles that are included in each sort of predictive state, you didn't actually need that many nodes, and you should have screened them and stopped updating them. And when you do that, you find that you have basically equivalent predictive capability, but you're much faster. Yeah, and in all cases that we looked at, so a lot of neuroscience were imaging at maybe 30 hertz for images, and EFIS is maybe up to 30 kilohertz, often binned. And so each of these we're doing sufficiently fast that we can do that, and the prediction ahead in time is going to be on the order of a second. So I think it's feasible.
Yeah, perfect. These are kind of like the considerations that I was hoping to get your perspective on. And
yeah.
Especially like what is also interesting is in terms of prediction, right? So like, does it make sense like to predict like for next step and kind of like always to follow in adaptive applications the next step, and then once you predicted it, you would act on it, or actually like to have also kind of like a long-term prediction that will kind of will in some sense will synchronize with the short-term prediction. What would be your take on these more conceptual kind of like questions and approach?
Yeah, so I think that's a really excellent thing to be thinking about. A lot of times what we consider to be sort of short timescales is the neural data, and longer timescales is the behavior. Usually task associated behavior, that's going to be taking place in the order of seconds or minutes, whereas the neural data is much faster. And so I'm actually working on building in the sort of behavioral correlates, or at least slowly varying things in time with our fastly varying things in time. But just looking at these actual predictions from a very noisy data set, we're well aligned with the trajectory to a certain extent. And then here our model actually says there's some split probability to go in either of these directions. And so we'll choose one of them, right? But we might be wrong on the long time horizon. We do better predicting on the short time horizon. But what's fun is to think about, okay, in these sort of bifurcation methods, which is a slower, we can see the event happening. How do we design stimuli to basically force it down, one path or the other? So I think being myopic, looking one step ahead in time is going to give us the truest predictions. And then figuring out how far ahead in time we're going to be uncertain about where we go. And then deliberately acquiring data that will make us more certain about the sort of branch points.
Excellent. Thanks so much.
Yeah.
Are there other questions for Anne? Going once, Going twice. And if not, then thank you again for a very interesting talk.
Yeah, thank you. And everyone should feel free to reach out or email if you're interested in talking more about this.